Molecular mechanism of gene expression. Multiple stage gene expression. Gene expression is a discrete stage in the pathway of information transfer from gene to gene product. It is a complex and multiple level molecular process completed in several sequential steps. The multiple stages of gene expression in eukaryotes include gene preparation, genetic transcription or mRNA synthesis, mRNA processing, mRNA export, mRNA turnover, genetic translation or protein synthesis, protein modification, protein targeting, and protein turnover. First, genetic preparation. Genetic transcription is usually considered as the first major step of gene expression. But before transcription, the gene has to be made accessible in a form ideal for transcription. This preparatory stage is very important in eukaryotes, where the efficiency of genetic transcription is enhanced by DNA methylation and the packaging of DNA in chromatin. The major events of gene preparation are the following: in viruses, uncoating, removal of capsid or protein coat, and the synthesis of template strand. In eukaryotes, modification of chromatin structure, changes in the topological and conformational properties of DNA methylation, genomic rearrangements, and gene amplification. Then genetic transcription. Transcription of the genetic code is accomplished by DNA-dependent RNA synthesis in nucleus using a DNA strand as a template. It is completed in 3 stages: initiation, elongation, and termination of mRNA synthesis. Then mRNA processing. This is the chemical tailoring or enzymatic modification of mRNA. It involves capping, tailing, Taylor silence, intron silencing, exon splicing, and mRNA editing. Capping is the addition of 7-methyl-guanosine at the 5' end of mRNA. Tailing is the addition of several adenine nucleotides at the 3' end. Intron silencing is the cleaving out of intron from mRNA. Exon splicing is the splicing together of exons. Then mRNA editing. mRNA editing is a genetic mechanism which involves the precise addition and deletion of nucleotides within the coding region of mRNA for modulating genetic activity. Then mRNA export. This is the export of processed mRNA from nucleus to definite protein synthetic destinations in the cytoplasm. This is the export of processed mRNA from nucleus to definite protein synthesizing destinations in the cytoplasm. Then mRNA turnover. This is mRNA degradation to its constituent subunits. Next, genetic translation. The mRNA-dependent biosynthesis of protein on cytoribosomes. It is completed in 3 stages: initiation, elongation, and termination. So, protein modification, post-translational chemical modification of nascent proteins. Genetic translation. The mRNA-dependent biosynthesis of protein on cytoribosomes. It is completed in 3 stages: initiation, elongation, and termination. Then protein modification, post-translational chemical modification of nascent proteins. This involves the cleavage of polypeptides, splicing of intermediate chains, folding of peptide chains to attain the final protein structure, chemical processing by complexing with chemical groups, and the interaction of modified protein with regulatory proteins. Then protein targeting. This is the process which directs nascent proteins to specific cellular locations such as nucleus, cell organelles, cellular secretion sites, etc. The signal hypothesis holds that the N-terminal amino acid sequence of a nascent polypeptide is very significant in protein targeting, especially in protein secretion. According to this hypothesis, the final cellular destination of a nascent protein is determined by a specific and relatively short amino acid sequence within the protein itself. It is called signal sequence, signal peptide, or leader sequence. Usually it consists of the first 13 to 36 amino acids of the N-terminal of growing polypeptide. It is believed to draw the ribosome towards the ER. It allows the transport of nascent protein to the appropriate cellular compartments by a process called protein trafficking or protein sorting. Signal peptide may be cleared out by the enzyme signal peptidase. When the protein reaches its ultimate cellular destination, a protein with the signal sequence that is sliced away later on is termed as pre-protein, and its signal sequence is called pre-sequence. Pre-protein differs from proprotein, which is a protein activated by proteolysis. The proprotein with activated pre-enzyme is called pre-proprotein. Signal sequence is a translation product when it is expressed on the ribosome surface. A cytosolic ribonucleoprotein particle called signal recognition particle SRP or docking protein binds to it. This blocks further mRNA translation until the growing polypeptide SRP or mRNA complex reaches ER and binds to it. SRP is a complex of 6 different proteins and 300 nucleotides long 7S RNA. Then protein turnover. This is the degradation and consequent loss of cellular proteins. Different cellular proteins differ widely from each other in their stability and half-life. Their half-life ranges from a few minutes, many regulatory proteins, to several weeks or more. Many structural proteins and storage proteins, collagen, hemoglobin, etc. These regulatory proteins are relatively unstable and they tend to turn over rapidly. Cells must have provisions to dispose of effective, damaged, and defective proteins. There are several protein degradation mechanisms or pathways in eukaryotes. Of this, ubiquitin dependent degradation and ubiquitin independent degradation are the important ones. The former is a proteasomal pathway taking place in proteasomes, where the later is a lysosomal pathway. Ubiquitin is a conserved protein with a highly conserved sequence of 76 amino acid residues. It is present in prokaryotes and eukaryotes as free molecule and also as conjugates with other proteins. Probably it plays a role in organization and regulation of chromatin and also in the regulation of cellular responses to stress. Ubiquitin is perhaps the slowest evolving protein identified so far. And we try to learn about major mechanisms involved in gene preparation level. Those include uncoating of nucleic acid in viruses, synthesis of template genome strand in viruses, modification of chromatin in eukaryotes, then changes in topological and conformational properties of DNA, changes in DNA methylation, genomic rearrangements, gene amplification. Then transcription, use of promoter, formation of active transcription complexes, then promoter escape, elongation versus pausing, then termination versus anti-termination in prokaryotes, then attenuation in prokaryotes. Then RNA processing in eukaryotes include capping, tailing or polyadenylation, and splicing of introns. Then RNA editing, addition and deletion of nucleotides in the coding region of mRNA. Then RNA export of mRNA from nucleus to cytoplasm. In eukaryotes, RNA targeting. Then RNA turnover, RNA degradation, retroregulation in prokaryotes. Then translation, ribosome binding, use of codon, frame shifting. Ribosome binding is initiation of protein synthesis or translation. Then protein modification, chemical modification, proteolysis, cleavage of polypeptide. Then some aspects related to splicing of introns, then adoption of quaternary structure, interaction with regulatory protein. Protein targeting include major mechanisms like targeting to cellular compartments, then secretion. Protein turnover include protein degradation and loss. Protein turnover, it is nothing else but the degradation and the consequent loss of cellular proteins. Proteosomes are large multifunctional enzyme complexes concerned with intracellular protein degradation. In fact, they are amalgamations of different kinds of proteases. They provide the principal mechanism for the intracellular degradation of the proteins which have outlived their usefulness or have become misfolded or damaged. Proteosomes are found in large numbers in archaebacteria, some eubacteria, and all eukaryotes. In bacteria, they are located in the cytoplasm, and in eukaryotes, they remain dispersed in the cytoplasm as well as in the nucleoplasm. So first major point is about genetic transcription. Transcription of the genetic code. Transcription is the transfer of stored information from DNA to RNA in the form of nucleotide sequence. So it is accomplished by DNA dependent RNA synthesis using DNA strand as the template. In this process, the mRNA copy of DNA sequence is produced as the first step of gene expression, making use of the base sequence of one DNA strand termed template strand or antisense strand. The whole process is mediated by RNA polymerase. First, transcriptional units or transcriptons. The elementary unit of transcription is known as transcripton. It is a small portion of a DNA molecule where actual transcription occurs. Only one strand of the transcripton is transcribed. It is known as the template strand, antisense strand, or anti-coding strand. The non-transcribing strand is called non-template strand, sense strand, or coding strand. The same strand of a DNA duplex may serve as the sense strand in one transcription and as the antisense strand in another. The RNA transcribed from an antisense strand is known as RNA transcript. It might be an exact replica or negative copy of the antisense strand and an identical copy of the sense strand, except that in it thymine is replaced by uracil. And RNA transcript is synthesized by the polymerization of ribonucleotide phosphatase, ATP, UTP, GTP, and CTP. During this, the 3' OH of one nucleotide reacts with the 3' OH of another, forming a phosphodiester bond. The sequence in which ribonucleotides are added to the growing RNA chain is determined by the sequence of the bases in the template strand. New nucleotides are added to the 3' end of the growing chain. This transcription always occurs in the 5' to 3' direction. Then transcription have some functionally difficult and different regions. In prokaryotes, they include the leader, promoter, operator, cistronic, terminator, and trailer regions. We learned this earlier. In eukaryotes, they include the leader, promoter, acceptor or regulator, cistronic, terminator, and trailer regions. So cistronic region consists of only exons in prokaryotes, but in eukaryotes it has alternating exons and introns. Only exons code for amino acids, so they are transcribed, translated, and genetically expressed. On the other hand, all other sequences are non-coding. So they are only transcribed but not translated and genetically expressed. When we try to learn about the prokaryotic transcription process, leader, promoter, operator, cistron, terminator, and trailer will be there. But eukaryotic transcription is like leader, promoter, acceptor, cistron, terminator, and trailer regions. Leader region plays some role in determining the rate of transcription. Leader region plays some role in determining the rate of transcription. Promoter region provides recognition signal to initiate transcription. In most eukaryotic RNA polymerase 2, it contains the CAAT box sequences and TATA box sequence. The former serves as a recognition site for RNA polymerase and the latter provides a binding site for enzyme. The binding of RNA polymerase to the TATA sequence initiates transcription. Receptor or prokaryotic operator region serves as the binding site of the regulator, which influence transcription. So leader region plays some important role in determining the rate of transcription. Then later, promoter region provides the recognition signal to initiate transcription. Then in most eukaryotic RNA polymerase 2, that portion of promoter contains CAAT box sequence and the TATA box sequence. The CAAT box serves as a recognition site for RNA polymerase and the TATA box provides a binding site for the enzyme. The binding of RNA polymerase to the TATA sequence initiates transcription. And next region is acceptor, we know that. So this acceptor or prokaryotic operator region serves as a binding site of regulators which influence transcription. The exons of the cistronic region contain the structural genes which code for amino acids and thereby provide the information for transcription. Then terminator region is there. So that terminator region provides the halt signal for the termination of transcription. Later, last portion is a trailer region. We know that. That trailer region is a non-translated sequence followed by a termination codon. Cistronic region is monocistronic in eukaryotes but polycistronic in prokaryotes. Then transcription enzymes are also very important. RNA synthesis is mediated by the enzyme RNA synthesis. So RNA synthase or RNA polymerase. It recognizes an appropriate promoter site in the antisense strand of the DNA transcription and binds with it. RNA polymerase recognizes an appropriate promoter site in the antisense strand of the DNA transcripton and binds with it. Then it mediates the synthesis of RNA using the antisense strand as a template. In prokaryotes, there are two kinds of RNA polymerase. One of them produces the RNA binding RNA primer during DNA replication, and the other one mediates the transcription of all the three kinds of RNA, mRNA, tRNA, and rRNA. In eukaryotes, there are three kinds of nuclear RNA polymerases in addition to the mitochondrial and chloroplast RNA polymerases. The three classes of nuclear RNA polymerases are polymerase 1, 2, and 3. The first one is located in the nucleolus and the others are localized in nucleoplasm. Polymerase 1 governs the transcription of all rRNAs other than 5S RNA. Polymerase 2 mediates the transcription of mRNA and some nuclear RNAs, and polymerase 3 is involved in the transcription of tRNA, 5S rRNA, and some other small RNAs. Bacterial RNA polymerase is a holoenzyme. It has two components, namely sigma factor and a core enzyme. The core polymerase is composed of 5 polypeptide subunits, namely 2 identical alpha subunits, 1 beta subunit, and 1 beta bar subunit, and 1 omega subunit. Only the holoenzyme can initiate genetic transcription. The sigma factor would be released soon after initiation. Then eukaryotic RNA polymerase are more complex. This is because there are about 10 kinds of subunits in them. The three classes of eukaryotic RNA polymerase 1, 2, and 3 differ from each other in their subunit composition. Some subunits are common for all of them, but the others are different. Mitochondrial and chloroplast RNA polymerases are very simple and they have only a single subunit each. Sigma factor has three major roles, namely it recognizes the polymerase recognition site of the promoter region of DNA template, enables the binding of enzyme to the binding site of the promoter, and it initiates RNA synthesis. On the other hand, core enzyme is concerned with RNA polymerization, elongation of RNA chain. The catalytic site of RNA polymerase is believed to be located in the beta subunit. Once the RNA chain has begun polymerization, the sigma factor dissociates from the enzyme and the core enzyme comes into action for chain elongation. Then transcription factors or TF. In addition to the RNA polymerase, some protein factors are also involved in mediating the transcription process. They are generally called transcription factors. They control the rate of transcription by binding to a specific DNA sequence around the data box. This binding forms a platform for the binding of RNA polymerase 2. Transcription factors are essential for the stable binding of RNA polymerase with the template strand of DNA, for the initiation of transcription at the correct codon, and also for the final release of RNA polymerase and the RNA transcript from the template strand. Genes without data box may have an initiator element. Still other genes have neither data box nor initiator element. Then components of transcriptional activity. Transcriptional activity is the rate of RNA synthesis. The number of full-length RNA transcripts synthesized per minute. Transcription initiation is usually rate limiting and also the primary level of gene regulation in prokaryotes and eukaryotes. So transcriptional activity directly reflects the efficiency of transcription initiation. The control of initiation involves three major components, namely basal components, constitutive components, and regulatory components. Basal components serve primarily to direct RNA polymerase to the start site of transcription, permitting the initiation of RNA synthesis. Then constitutive components control the efficiency of initiator of in the absence of regulation and allow the expression of different genes at different rates to meet the basic needs of the cell. Then regulatory components alter initiation efficiency, allowing the modulation of transcriptional activity of individual genes in response to changes in the environment. Then transcription in prokaryotes. In prokaryotes, transcription is completed in three major stages, namely initiation, elongation, and termination. So initiation of transcription involves all the molecular events occurring before the incorporation of the first nucleotide. It is completed in three main steps as follows: binding of the holo enzyme RNA polymerase with the promoter region of DNA, local unwinding of the companion strands of DNA. This is induced by RNA polymerase, which has both DNA unwinding and DNA rewinding powers. Local unwinding makes the template strand free for base pairing with free ribonucleotides. Phosphodiester bonding between newly incorporated initial ribonucleotides. Then during initiation, the sigma factor of RNA polymerase recognizes a specific sequence in the promoter region of the template strand known as recognition site. This may be called template recognition. Soon, sigma factor complexes with the core enzyme and forms the RNA polymerase holoenzyme. The holoenzyme interacts with the binding site in the promoter region, then moves to the operator region, and finally comes to the initiation site in the cistronic region to initiate transcription. Transcription usually begins at the initiation site. Sigma factor has a significant role. It regulates the promoter region and ensures that RNA polymerase binds exactly with the binding site. It can bind with the binding site only when it is complexed with the core enzyme. The core enzyme cannot exactly recognize the promoter sequence all by itself, and so it cannot initiate transcription without complexing with the sigma factor. The binding of the sigma factor of RNA polymerase with the promoter region causes the local melting or disruption of hydrogen bonds and the unwinding and separation of companion strands of DNA duplex. The unwound region of the transcripton is sometimes called transcription bubble. Here we are talking about transcription in prokaryotes. It moves forward along with the polymerase. To avoid strain on the adjacent intact region on the DNA double helix, only a short region or about 12 to 17 BP base pair is unwound at a time. Soon one strand of the bubble acts as a template strand and directs synthesis of an mRNA on it. Once the mRNA chain has become a no nucleotide long, usually 2 to 10, sigma factor is no longer required, so it detaches from the core enzyme and the DNA template and becomes free to get complexed with another core enzyme to form yet another holoenzyme. At the same time, the core enzyme comes into action, moves forward, and brings about chain elongation. Then elongation process in prokaryotes. Elongation or polymerization of mRNA chain occurs within the transcription bubble soon after the release of the sigma subunit. It is catalyzed by the core enzyme of RNA polymerase, which keeps on moving along the template strand in the 3' to 5' direction. As the core enzyme moves forward, it unwinds the intact DNA strands ahead of it and at the same time rewinds the separated strands behind it, leaving the transcription bubble in between. Soon, activated ribonucleotide phosphatase like ATP, GTP, CTP, and UTP covalently bind to the corresponding complementary nucleotides of the template strand through hydrogen bonding. Adjacent nucleotides are then linked together through energy-rich phosphodiester bonds with the help of core enzyme. The energy required for this is made available by the hydrolysis of terminal bonds of nucleotide phosphatase. Chain elongation always occurs in the 5' 3' direction. The binding of DNA polymerase to DNA and growing mRNA maintains the stability of transcription complex. RNA polymerase has no proofreading powers for correcting mistakes and repairing damages. Therefore, the incorporation of ribonucleotides during chain elongation must be absolutely error-free. Otherwise, there is high probability for mismatch binding and copy errors. Elongation of the mRNA chain initially forms a transient and very short DNA mRNA hybrid or heteroduplex. Soon, the 5' end of the mRNA strand progressively separates from the DNA strands. As a result, the template DNA strand becomes free from mRNA transcript, and two DNA strands reassociate to form DNA duplex. Then next, termination. Termination of transcription occurs at a specific DNA sequence which serves as a stop signal for RNA polymerase. When the core enzyme of the RNA polymerase reaches this sequence, the transcription bubble collapses and the transcription complex dissociates. The core enzyme and the mRNA transcript are released and the DNA returns to its double helical state. Then transcription termination involves three major events, namely cessation of polymerase movement and termination of chain elongation at the terminator sequence, release of the mRNA transcript from the core enzyme, then dissociation of core enzyme from the DNA template. The released core enzyme will soon bind to segment in the DNA and will move to the promoter for complexing with the sigma factor. Then there are different types of termination based on the involvement of chain terminating protein called Rho factor. Two kinds of transcription termination can be recognized among prokaryotes, namely Rho independent and Rho dependent. The functional form of Rho is a hectamer with 6 identical copies. Rho independent termination can occur even if Rho is not present. On the other hand, Rho dependent termination occurs only when Rho is present. In Rho independent termination, RNA polymerase continues to mediate transcription past the terminator sequence by a process called read-through. Then Rho independent termination. Rho independent termination is also called intrinsic termination. It is the built-in mechanism in which the transcribed mRNA transcript causes and contributes to the termination of its elongation. In this case, the stop signal is provided by a specific termination sequence or terminator sequence on the mRNA transcript without the preparation of Rho factor. The core enzyme recognizes the signal sequence and stops moving beyond it. As a result, mRNA polymerization stops. In some instances, there will be special transcriptome terminator sequences called attenuator sequences, which may cause optional termination. They permit transcription to continue when the gene product is insufficient and terminate transcription when there is enough gene product. Then Rho independent termination, we learned about that later. Rho independent termination. The Rho dependent termination. We just learned about Rho independent termination. Now we are going to learn about Rho dependent termination. In Rho dependent termination, Rho factor plays an active role. It first recognizes the stop codon in the template's DNA, then it binds with the core enzyme when the enzyme reaches the terminator sequence. This Rho factor core enzyme complexing brings the enzyme to a halt and disrupts the base pairing between the DNA template and the mRNA transcript. Also, it causes the detachment of the mRNA from the DNA and the enzyme and the release of core enzyme from template DNA. This in turn terminates transcription. The mRNA transcript released from the enzyme is known variously as heterogeneous nuclear DNA, hnRNA, nuclear RNA, hnRNA, primary mRNA, precursor mRNA or pre-mRNA. Then transcription termination through stem loop configuration. In bacteria, Rho independent and Rho dependent terminations of transcription occur at a specific sequence in the mRNA transcript known as inverted repeat sequence. In it, the first half of the sequence is followed by its inverted repeat sequence in the second half, with a short sequence in between. Just before the first base is transcribed, the inverted repeat sequence assumes a hairpin-like stem loop configuration in the mRNA strand through intramolecular base pairing. The stem loop structure acts as a signal for termination. Also, it enables the detachment of mRNA transcript from the template strand of DNA. Next topic, eukaryotic transcription. Transcription in eukaryotes. Eukaryotic transcription is almost similar to that of prokaryotes. However, it differs from prokaryotic transcription in the following respects. There are three types of RNA polymerases 1, 2, and 3. Only one type in prokaryotes. mRNA synthesis is catalyzed by RNA polymerase 2. There are three different types of promoters for the three classes of RNA polymerase. Only one type of promoters in prokaryotes. Then initiation of transcription is more complex than in prokaryotes. Termination of transcription does not involve stem loop structure. Stem loop is present in prokaryotes. Binding of RNA polymerase 2 to its promoter involves a number of proteins called transcription factors. No transcription factors in prokaryotes. Then nearly 50% or more of the promoters contain a specific sequence TATAT called Tata box or Goldberg-Hognes box, absent in prokaryotes. In some genes, other sequences such as CAAT box may be present beyond the TATA box, absent in prokaryotes. They act as binding sites for the transcription factors which stimulate or repress transcription initiation. Many genes may contain sequences such as enhancers and silencers located outside the promoter, absent in prokaryotes. Enhancers induce and silencers inhibit transcription. Then termination of transcription in eukaryotes occurs at a point much beyond the end of protein coding sequence. The actual mechanism of termination is not definitely understood. It is generally held that definite termination signals do not exist in eukaryotes. At the same time, dissociation of a transcription factor at some point is believed to destabilize the transcription complex. This, in turn, causes the detachment of RNA polymerase 2 from the template strand, resulting in the termination of transcription. Then, transcription complex. The major components of eukaryotic transcription complex include RNA polymerases, transcription factors, promoters, enhancers, and silencers. RNA polymerases in prokaryotes, there is only a single kind of RNA polymerase, but in prokaryotes, in case of eukaryotes, there are three classes of structurally and functionally different kinds of RNA polymerases 1, 2, and 3. RNA polymerase 1 is located in nucleolus and it displays the transcription of class 1 nuclear genes to synthesize all types of rRNAs other than 5S rRNA. DNA polymerase 2 is located in nucleoplasm and it governs the transcription of class 2 nuclear genes, which include all protein coding genes and some genes which code for small nuclear RNAs. RNA polymerase 3 is also located in nucleoplasm and it mediates the transcription of class 3 nuclear genes to synthesize tRNA and 5S RNA. Then transcription initiation factors. TIFs, also termed basal transcription factors, BTFs, or general transcription factors, are the accessory protein factors which assist RNA polymerase to recognize their promoter. They bind with the DNA and form a complex which recruits RNA polymerase to the transcription initiation site of the DNA. The TIFs and the enzyme collectively form the pre-initiation complex which forms the basal apparatus of transcription initiation. Then eukaryotic promoters, enhancers, and silencers. So eukaryotic promoters, enhancers, and silencers are cis-acting regulatory sequences. They are capable of local interactions with the pre-transcription initiation complex in eukaryotes. In bacteria, they interact with RNA polymerase. In eukaryotes, constitutive transcription regulators usually bind up at sites upstream from the basal promoter. In bacteria, they flank the promoter. Enhancers and silencers are distant regulators located outside the promoter, hundreds or thousands of base pairs away from the genes they control. Enhancers are positive regulators which stimulate transcription, and silencers are negative regulators which inhibit transcription. They are quite common among eukaryotes but rare in prokaryotes. Next, eukaryotic promoters. Promoters are cis-acting control sites. proximal to the genes they control. A promoter consists of two components, namely basal promoter and upstream promoter. RNA polymerase binds with the former, or the basal promoter. The latter contains constitutive and regulatory elements. Many genes contain, so we learned about promoters, enhancers, and silencers, and you are learning about eukaryotic promoters. Promoters are cis-acting control sites. Cis-acting sequence proximal to the genes they control. The portion of RNA polymerase 1 is species specific in the sense that RNA polymerase of a particular species can bind only with the promoter of that species. The promoter of RNA polymerase 2 in most cases may contain Tata box and CAAT box. The Tata box or Goldberg-Huggins box is a eukaryotic promoter element to which RNA polymerase binds during transcription. This is usually a 6 or 7 base pair long nucleotide sequence located 19 to 27 base pair upstream from the start point of eukaryotic structural gene. Tata box is analogous to the Pribnow box of prokaryotic sigma subunit of RNA polymerase binds during transcription. This is a 6 or 7 base pair long segment located nearly 10 base pair upstream from the start point of transcription site. The commonest Pribnow sequence is TATAAT or TATAATG. CAAT box is a common promoter sequence of many eukaryotic genes that are transcribed by RNA polymerase 2. It is found upstream of Tata box. It probably plays an important role in promoter function and is necessary for efficient initiation of transcription. Then eukaryotic enhancers. Enhancers are short cis-acting and distantly located positive gene regulators, positively acting control sites, which can enhance transcriptional activity. Quite often they contain both constitutive and regulatory elements which can increase transcriptional activity up to 1000 times from very great distance. Enhancers do not show promoter activity because they lack basal components. They may contain functionally clustered groups of binding sites termed enhancons. Many transcription enhancers contain the conserved octameric sequence ATTGCAT in which some transcription factors interact. Some enhancers are constitutively expressed in most types of cells, while the others are tissue specific or species specific. Enhancers act by increasing the number of RNA polymerase molecule. Their enhancer activity is mediated by species specific DNA binding proteins. The attachment of DNA binding protein to an enhancer causes the looping out of nucleotides between the enhancer and the promoter. This looping brings the enhancer into physical contact with the promoter of the gene it enhances. The loop structure then facilitates the binding of RNA polymerase with the transcribing gene. Then eukaryotic silencers. Silencer sequences are short, distantly located and cis-acting negative regulators or negative control sites. They are common in eukaryotes but rare in prokaryotes. They are associated with the genes that are transcribed under the control of delta 54 factors. Silencers may be located upstream or downstream of the genes or rarely within the genes they regulate. They can bind with constitutive and regulatory transcription factors and interact with basal initiation complex to control transcription initiation. Silencers exert a negative effect on the rate of genetic transcription. They express promoter activity in those cell types which other than their target cells. So they are cell type specific negative transcription regulators, or in brief, cell type specific transcription inhibitors. Then cis-acting DNA sequences. Cis-acting elements are short eukaryotic DNA nucleotide sequence which can regulate the genes that are seen on the same DNA or chromosome on which they are located. The genes in the cis configuration without any effect on the genes on other DNA or chromosomes. Genes in the trans configuration. A cis-acting element usually consists of 10 to 12 base pairs and is located either within the gene or within the 5' flanking region. Cis-acting sequences represent the recognition site with which specific transcription factors interact. They are essential for eukaryotic gene regulation. In general, they do not code for proteins but rather serve as attachment sites for DNA binding proteins. Enhancers, silencers, promoters, and operators are examples of cis-acting sequences. The cis-acting elements in general regulate genetic transcription, RNA processing, and protein synthesis. Then, stages of eukaryotic transcription. Then, just as in prokaryotes, genetic transcription is completed in 3 stages in eukaryotes also. They are initiation, elongation, and termination. During initiation, RNA polymerase II binds to the promoter site of the DNA template and initiates RNA synthesis. During elongation, the RNA transcript extends to the desired length by the incorporation of more and more ribonucleotides, and during termination, RNA synthesis stops and the RNA transcript dissociates from the DNA template. Then initiation, the transcription of mRNA begins with the binding of RNA polymerase II with the promoter of DNA template. The promoter is located close to the transcriptional start site of the protein coding gene. Transcription initiation occurring in those promoters with TATA box or TATA box is slightly different from that occurring in TATA-less promoters. In transcription initiation, TATA box serves as a recognition site for several transcription factors. Also, it plays an important role in positioning the initiation complex exactly at the very start site with anchoring precision. In this case, the first step in initiation is the recognition of the TATA box by some transcription initiation factors such as TATA binding proteins and a variety of TEB associated factors. TEB makes sequence specific contacts with the DNA. TAFs recognize known TATA elements and the transcription initiator of the core promoter. They also allow all transcription initiation factors to respond to transcriptional activators and silencers. Thus, the TATA box is activated by recognition and binding by DNA polymerase II. Quite a number of promoters are TATA-less, yet they can initiate transcription. In them, transcription initiation is believed to be controlled by a CT-rich sequence called initiator element in association with the downstream promoter element or DPE. INR or that initiator element is located very close to the transcription start state and DPE is located 28 to 34 base pair away. The binding of RNA polymerase II with the promoter generates a closed promoter complex. In successful transcription, it gets converted into an open promoter complex in which the DNA remains locally unbound. Now, transcription begins with the incorporation of the first ribonucleotide mediated by RNA polymerase II in association with the transcription initiation factors. Soon the enzyme moves forward from the promoter site without dissociating from the DNA transcripton and at the same time freeing the promoter for further initiation. This is called promoter clearance and it marks the end of transcription initiation. Promoter clearance occurs only when the open promoter complex is stable. Usually it follows several abortive initiations in which only short transcripts are generated. Then elongation. Once successful initiation has been completed, RNA polymerase II moves forward along the DNA template, catalyzing the incorporation of more and more nucleotides and synthesizing an nascent mRNA in the 5' to 3' direction. This brings about elongation. During this, RNA polymerase II gets associated with a series of transcription elongation factors which prevent its dissociation from DNA template until it reaches the end of the protein coding gene and elongation is over. In eukaryotes, transcription elongation is always coupled with the co-transcriptional DNA RNA modification. During transcription elongation, the double-stranded DNA gets unwound ahead of the elongation complex and rewound behind it. This results in a transcription bubble with the disruption of nucleosome organization of DNA. Then termination. The termination of eukaryotic mRNA transcription is only poorly understood. It involves the dissociation of enzyme and the elongation factors from DNA and the liberation of nascent mRNA transcript. Then in eukaryotes, termination of transcription by mRNA polymerase II occurs at a point beyond the end of protein coding gene. It is believed that transcription termination probably occurs when RNA polymerase encounters either a chemical impediment or receives a stop signal from termination codon of DNA template. However, no such termination signals have so far been identified. An alternative view is that termination signals are altogether absent, and dissociation of the elongation factors at some point destabilizes the transcription complex. This in turn leads to the dissociation of RNA polymerase II, termination of transcription, and release of mRNA transcript. Thank you for listening.